So this is Chapter 17, Part A. It's going to be about the specific defenses. And this is going to be when the nonspecific inflammation and phagocytosis does not work. And then you have to rely on the next level, which is going to be the level of specific immune response because of the involvement of lymphocytes. So when you're talking about specific defenses, it's going to address just the pathogen that the immune system is having problems resolving, getting rid of. So we've already done innate or nonspecific immunity. And that means that when you're doing inflammation and phagocytosis, you're going to deal with you're going to deal with any pathogen the same way. In other words, whether it's a virus or it's a bacteria, you're going to do inflammation, you're going to do phagocytosis. Then, if that does not resolve the problem, then and only then do you go to the next specific line, which we're going to start to talk about today because it's divided into two parts. We're mainly going to be talking about B cells and antibodies, and then the next chapter will be T cells and natural killer cells and T cytotoxic cells, blah, blah. So we know that immunity is a specific response now to an antigen, so a specific antibody and lymphocyte response to an antigen. So what is an antigen? Now, this definition says a substance that causes the body to produce specific antibodies to sensitize T cells. I'm going to give you another definition. It's going to be molecules on the surface of cells that allow the immune system to see them as foreign or different. So an antigen is a molecule that is seen by the immune system as foreign or different. The immune system, when you are a fetus, memorizes every cell in the body, every tissue, every organ system, and labels it as self, S-E-L-F, self. Anything else is considered non-self. So all pathogens carry some non-self antigenic molecules that the immune system can recognize as foreign or different, and then resolve to eliminate them. An antibody is one type of response. So an antibody is a Y-shaped protein made in response to an antigen. Now, it's also called an immunoglobulin, which is abbreviated capital I, small g. They are the same thing. It's one of those situations which is common in medicine and science where you have two different fields and they come across this Y-shaped protein, and if you are into serology, you would call it an immunoglobulin, and if you are into immunology, you would call it an antibody. They're exactly the same thing, but you can't get a serologist to call that Y-shaped molecule an antibody any more than you can get an immunologist who would prefer calling it an immunoglobulin. They're the same thing. Lucky you as students, you get to memorize both names, and they mean this Y-shaped antibody that can bind to an antigen and lead to its destruction. Okay, next slide. We are talking about acquired immunity, meaning that you're not born with it. You acquire it as you live your life and you come in contact with environmental pathogens and they try to invade your body. And that's the reason why during your lifetime you can develop memory. You survive to tell the tale, you evoke that third line of defense, that B cell and T cell, and as a reward, so to speak, for living to tell the tale, then ta-da, you have acquired immunological memory. It comes in two flavors. Immunological memory comes into, or acquired immunity, comes as A, humoral immunity, and B, cell-mediated immunity. The lecture on this Chapter 17a is all about humoral immunity. humoral, old time term, the humors of the body, and the blood was one of the humors of the body. So since antibodies are found in the blood, they got the nickname humoral immunity. It involves antibodies that are produced by B lymphocytes. Apparently, people got really tired of saying lymphocytes, so a B lymphocyte got nicknamed a B cell. Cell-mediated immunity, as the name implies, is going to be about cells, specifically T lymphocytes or T cells. But that's going to be the Chapter 17b lecture. Today it's all about antibodies. Okay, acquired immunity. Let me go through this quickly because a couple questions could be on the quiz, but in actuality, this is where we're going to talk a little bit about vaccines. You can acquire natural active immunity, get the disease, live to tell the tale. There you go. Then you have acquired a very active immunity. You always have a certain level of the memory antibodies to interact so that you can be exposed, but you won't get the disease again. Now, natural acquired passive immunity is going to be something that, now they say transplacental or via colostrum. I would also include mother's milk, breast milk, because when you breastfeed your baby, you already have these memories. You have memories of being vaccinated. You have immunological memory of getting a disease and recovering from it. The immunity you got from vaccinations in your lifetime and the immunity from diseases that you fought in a lifetime, you can get secreted in mother's milk. There's a particular type of antibody called immunoglobulin A that is present. And so when you breastfeed your baby, until you stop breastfeeding, that child is acquiring your immunity. And it's natural. So naturally acquired passive immunity. Passive why? Because when you stop breastfeeding the baby, then the child's immune system is on its own. And that's usually when you start to vaccinate them and protect them in that way. Mother's antibodies only last about a week or so that when you stop breastfeeding the baby, then the baby cannot, from your antibodies, from your immunological history, the baby cannot actively acquire it. It passively takes it, but when you stop breastfeeding, then they're on their own. So that's the reason for artificial acquired active immunity. You vaccinate the child. And artificial, because vaccinations are not natural. Chemical, pharmaceutical companies make them. And it, however, is based on exposure to an inactivated pathogenic antigen that will then trick the immune system into thinking it's real so that you get memory, but you don't have the actual disease. So childhood killers like chickenpox and polio and measles and mumps and etc. A child in the 21st century doesn't have to be exposed to, they can be exposed to those, but they are vaccinated so they won't get sick. And then last but not least, what happens to adults if they are suddenly exposed? And vaccinations takes about 6 weeks to actually be sure that but what if all of a sudden you are exposed and you need immediate immunity? Yes, you can get it. It's called artificial acquired passive immunity. An injection of an antibody. Usually you're injected with what is called gamma globulin, which are an enriched source of antibodies to a specific pathogen. So I get to tell my story. So I'm in grad school and I have a wonderful dissertation advisor, but he, unbeknownst to him, came down with infectious hepatitis type A. And one of the ways that it is acquired is through secretions like saliva. And he didn't realize it, and he actually, drank from a punch bowl and then found out that he had it. And because of the way that he drank from the punch bowl, meaning that he didn't rinse out the cup, but he just put it back, he then came in a few days later and said, okay, everybody in the lab, well, you're going to have to go down and get a gamma globulin shot against hepatitis A. He did this by a phone call because that's what I have, and I think I might have contaminated the punch bowl. So we all ran down to the health center and got our gamma globulin shots, and this all has a happy ending because it's infectious. It's not a dangerous disease. He recovers. We got the gamma globulin shots that was enriched for hepatitis A immunity or antibodies, and we were fine. But that's an example. So very often if people are exposed, a subpopulation gets exposed to something that gamma globulin has been or is available, then those people need that within a very short period of time. It might be within 24 hours, 48 hours. Otherwise, they'll come down with the disease. Now, a vaccination is going to take 6 weeks. So that person has to have an artificially acquired passive immunity. They have to have a gamma globulin shot. Okay, so this is just showing you. These are, without getting into a lot of the verbiage of all of this, these are the types of molecules that could be seen as non-self or as antigen. And this is that Y-shaped antibody that at the tip of the Y has an antigen binding site. And then it's able to specifically bind to that bacteria or virally infected cell, and in doing so starts a cascade in which the cell will be destroyed. Okay, antibodies come in humans in 5 flavors: IgG, IgA, IgM, IgD, and IgE. I'm only going to cover 4 because those are the 4 that are more prevalent in either vaccines or in fighting disease in different parts of the body. So one of the most prevalent type of antibodies is called IgG, immunoglobulin G. They are named after the Greek alphabet. So G is for gamma. I'm not going to test you on this. A is for alpha, E is for epsilon, D is for delta, etc. So if I am a serologist, I would talk about IgG antibodies, and because all of the 5 types that humans make are called immunoglobulins G, A, D, M, or E, even immunologists have to call them by that name as well. So here's my little Y-shaped molecule that I would call an antibody because I'm more classically trained as an immunologist. And then the tips of the Y-shaped molecule is where you would have very specific binding to a very specific pathogen. So you only have one of them at a time, so they're called monomers. Most, 80% of all antibodies in the blood are IgG. They can enhance phagocytosis, they can neutralize toxins and viruses, they can also bind to bacterial and fungal cells and start a cascade that would lead to their destruction as well. This can go past the placenta and protect the fetus in utero, and the newborn is protected if the baby is fed almost exclusively breast milk. All right, IgM is the biggest of all. There's a chain that holds them together, kind of like a keychain. So you have 1, 2, 3, 4, 5. So that means this huge IgM molecule, although you only have 5 to 10% in the serum, it's kind of like the first out because it can just spin around and capture so many pathogens. And then the next wave will be IgG. So that's why it says It agglutinates microbes. First, antibody produced in response to an infection. So you're going to basically get the heavy guns out there first, kind of followed by the infantry second. So IgM is first, and then IgG. All right, you have parts of your body which are also under attack. So IgA is going to be the antibody of body secretions. So it is a dimer. There are two of them held together by a secretory component and a J chain. And the whole secretory component is needed for it to secrete into mucus, and to secrete into breast milk, to secrete into sweat, to secrete into tears. And that way, things that might affect your eye, things that might affect your mucous membranes, things that might affect your skin, and obviously secreted into breast milk to protect to protect the newborn baby, is all about IgA. So you find them, very little of them in serum. In fact, if they're in the serum, they're on their way to be pushed into a secretory compartment. So it is the antibodies to protect parts of the body where body secretions occur, like sweat, like tears, like mucus, and obviously mucosal protection. And then last but not least, IgE antibodies, the antibody of allergies. If you don't have allergies, the amount of serum antibodies, look at it, 0.002%. I'm allergic to summertime. I'm allergic to pollen from the trees, ragweed, all kinds of stuff like that. So, Flonase is my friend. What can I say? So if you took my IgE and titered it or found out its percentage in my blood, it might be right about now 20%. And then in the winter, when the deciduous trees die and the plants die, then I could be 0.002%. And then comes the summer. And so it really kind of depends. People who are asthmatics very often have IgE triggers. So they have to figure out what the trigger is. What is the allergy trigger? My sister, for example, is allergic to strawberries, and when she eats them, she throws up and she has diarrhea, and that has to do with the IgE in her gut. So it kind of depends on what you're allergic to. Could be pollen, could be dust, could be food, etc. And then if it's respiratory, so I have a friend who is allergic to dog dander. So if she's around dogs, she gets very wheezy and has to take her inhaler and things of that nature. So IgE, however, would be suddenly quite high. And because it's a memory antibody, it can become quite high quite quickly. So she has to stay away from dogs because that's her trigger for an asthmatic attack that is propagated or initiated by IgE. So that's a lot on IgE, but because we have so many children allergic to peanuts, and we will get into these adverse immune responses and talk about peanut allergies later on, but it's so prevalent in so many children now that knowing about IgE and the antibody of allergy is important. And also to realize that if you take a blood sample of a child right after an asthmatic attack, their IgE is going to be out of sight. If the child next to them is not allergic to anything, that's when you see the 0.002%. You're going to find that they have binding sites on mast cells and basophils, both of which can produce histamine. And that does bring about an inflammatory response. And now, in the olden days, when you had people in very unhygienic circumstances, where you had a lot of food be infected by parasites, especially parasitic worms, then IgE was very good at expelling or starting a diarrheal or dysentery response, which would purge the person's intestines of worms. Nowadays, we live in a hygienic society and we don't have to go around purging worms, but the downside of it is that allergies are much more prevalent. Asthma is much more prevalent. And it's all mediated by IgE. And that is it. So what will happen in the next segment is we'll talk about T lymphocyte response. So we've talked about B lymphocyte, which produces antibodies, and how antibodies get involved in the immune response. And now we're off and away to T lymphocytes. See you then.